You can start.
Okay. Hi everyone. In this opportunity, I will explain about our work. This is in silico screening of commercial drugs like compounds for covalent inhibition of Tc80.
Please speak louder because we don't hear you.
Okay, sorry. Well, the Chagas disease is an endemic tropical illness prevalent in Latin America. This is caused by trypanosome cruzi, and one of the proteins that T-cruci has is TC80, a serine protease enzyme that is involved in parasite maturation and invasion. So we found in different kind of studies that 1A compound is from like a TC80 selective covalent inhibitor. So for that, we use this compound like a model to find another compounds to prove the activity with that TC80 protein. The objective of the study is just study the covalent inhibition potential of commercial drugs against TCA3 through in silico screening with the goal of identifying promising candidates for the treatment of Chagas disease. The methodology was first found a different kinds of database of compounds, and then we pass it with a Warhead filter. After that, we use the F3 similarity to filter again, and after that, the structural activity filter, and finally the toxicity and ADME filter. With that, we found a short number of compounds that then will be proved in the covalent molecular docking with that structure or the 3D structure of the protein that is the TC80. So that in this part, I want to show what we do, what we did. First, we start with 76,496 covalent compounds, and after all the filters, we finish with 121 compounds. About the TCAT 3D model, we need to know that TCAT doesn't have a 3D model. For that, we need to do the homology modeling, and we did that, and we found this modeling, and also the site active. And I also show the Ramachandra plot to show us that 3D model was successful. Okay, so then after that docking, we found this compound, the 1794 compound that was found the best compound of all the products because it has affinity in the micromolarity numbers instead the 1A compound that was in the millimolarity numbers. And in this, I want to show you the structures and also the bonds with the proteins. Compare the 1A and also the compounds 1694. So finally, in conclusions, we obtained promising compounds with better estimate affinity than 1A, highlighting compound C1694 that present multiple interactions with residues of the active site, including a hydrogen bond with the residue serve 548 of the catalytic field of Tc80. So to finish, I want to, well, my partners and I, especially thank Biosolve for giving us the opportunity to participate in the contest and win the Caesar software license. And also a special thanks to the Chemistry Students Society for Research for supporting that work. Thank you.
Thank you for your presentation. And we have one question from Vladimir Sulimov. Do you use any structure of TC80 from Protein Data Bank? What program do you use for covalent docking?
For the covalent docking, we use the Caesar software, as I showed here. That's a question? Or...
What about the records from Protein Data Bank?
Sorry, can you repeat the question?
How do you construct 3D model of the target protein?
Okay. First, we search in Protein Data Bank the sequence of the protein Tc80, and then we pass it with different kind of softwares to finally found that homology model that I showed that.
Do you use homology to construct the model?
We use Swiss model and also Galaxy Web to do that.
Okay, I understand that there are no PDB structures of this protein.
There are. There isn't. Tc80 doesn't have the 3D structure.
And what docking program do you use to perform covalent docking?
The Caesar software of Biosolve. We use that to do the covalent molecular docking.
Okay, thank you.
Okay, thank you. We have one question from Dmitry Shulga. Why you adhered to covalent binding in designing new inhibitors?
Why we use a warhead? Or what kind of software?
Why you would like to find covalent inhibitors, I think.
Covalent inhibitors, okay. Because they are more stronger the affinity is stronger than no covalent inhibitors. And also because there are studies that show us that the interaction between compounds and Tc80 is with covalent. It's like covalent.